ahead and get started. My name is Joan Dalusung. I'm the manager of the Paseo Verde Library here. And we are in the middle of our summer reading program, which we have for all ages. You're welcome to sign up after the program if you want to. Every year there's a theme. So this year's theme is science. So we all try to do science programming. And so we have this opportunity to welcome folks from the Desert Research Institute. We have Schell, their Public Information Officer, and then Dr. Scott Hamilton-Brehm, who will be giving the main presentation tonight. So please help me welcome them.
Before Scott gives his talk, which I think you'll find really, really interesting, I just wanted to let you know a little bit about DRI. Who here has never heard of DRI before, the Desert Research Institute? Everybody else has a pretty familiar understanding of our research and what we do? Yeah, so we're doing research on every continent, including Antarctica. So from studying water quality to looking at the dynamics of climate change, to invasive species in Lake Mead, to some of Scott's fascinating research with the NASA Astrobiology Institute project. We're doing a wide array of things. So feel free to reach out to us at any time. We hold events throughout the year, and one thing you guys might not be familiar with is our Green Power Education program. So what we're doing is we're taking the science that we're doing on these very high levels, and we're putting that curriculum into the classroom. So these science kits are delivered for free to classrooms across the state of Nevada and other educational resource entities like the libraries or other things like that. So you, from kindergarten to 12th grade, you can check out a green box, and from everything from hydroponics to geospatial imaging to having kids learn how to make solar panels, we do it all. The resources are free, and teachers love it because it's delivered to the classroom, and when they're done with it, we pick it up and it's delivered back to DRI where it's restocked. So I have information about that here, and you can come and talk to me after the program about that. And I hope you enjoy the talk. It's going to be really, really fascinating. Thank you.
Hi. Yes, so my name is Scott Hamilton-Brehm, and I'm a senior postdoc at the Desert Research Institute. I don't know if you're not familiar with the PhD and the doctorate programs. You might hear like medical doctors do residencies in hospitals. So they're doctors, but they have to kind of prove their skills and build up new skills. In the PhD program, we do postdocs, so the same thing. So this is my second postdoc, and my background has always been with biochemistry, but I've kind of slipped over to microbiology, and I specialize in working with odd and unusual chemistries in organisms, which is going to be the basis of my talk here. I have some little built-in stopping points here, so I will stop there and you can ask questions, so you don't have to necessarily remember things from the beginning and have to wait till the very end to ask questions on it, but I'll let you know when we hit those stopping points. So. All right, so ready to go? So, Earth, Mars, and microbes. This is Earth, as seen from Saturn. Pale blue dot. Now, it doesn't seem too much this far out, but when you get a little closer, Earth is a water world. It has clouds, land masses, and it's a home for a large variety of different organisms, from the large and great, like whales, to the humble honeybees. Lots of different varieties of structures, such as, like, you've got scorpions out here. But not just the present. In the past, the dinosaurs, again, massive creatures that walked on this planet. And there's always us, the intelligent life form on the planet. But there's also different terrains, like the Africa, the belts, where you have the elephants. And then even in the very Arctic, cold environments, and you have Siberian huskies. But these are all the organisms you normally encounter and think about all the time, but there's another one. Another organism, a life form on this planet, and it constitutes most of all the biomass on this planet, but you never see it. This organism you can only see with a microscope. When you look down into a water sample, you see this. Lots of them. What are they? They were considered little animals initially, but we later find out that we call them microbes or bacteria. And like I said, they dominate every corner on this planet. There's not one spot you can look without finding them. So, what are they? What do they do? Well, they're almost like the invisible engineers of this planet. You look in you see green slimes on it. These are the cyanobacteria. They produce gases, oxygen. They convert lots of chemistries on this planet for us. We even use them to make food. And they also take food away too. We make beverages out of them, and of course we do know what we do know of them. We don't think, oh, disease. They're bad. They're bad. Yogurts, cheeses, but ultimately, whenever you talk about microbes, you always think about, oh, they're bad. They cause disease, but this is only a fraction of what they are. Let's start off with us. We think. We talk about disease, but really, this is an up-and-coming new science where we're finding out that there is more of them than there is us. At least 10 times, there are 10 times more of their cells than us. So we're just a scaffolding, actually, for these microbes, and they actually are very beneficial to us. This is like I said, an up-and-coming research. We're finding out that our physical health, even our mental health, is controlled to a certain degree by them. So it may be in the future where you may go to a doctor, and it may not just be a simple matter of, let's give you some antibiotics, or let's kill off the microbes that are in you. They may actually give you a supplement saying, we need to bolster and boost the organisms that are good for you. We need to have them take care of the problem for you. Like I said, right now, the main thing is that they're finding out that our microbes wage a considerable war against other microbes that we come into contact with, because they like it being with us. We're a good home to them. Why let an invader come in? Usually when you get food poisoning, it's not like that means an invader got in. And so, in conjunction with your immune system and them in your system, they'll fight it off. So like I said, expect a lot of research in this area. It's really interesting, fun stuff. So, what else? Why are microbes? Why are you studying microbes? Well, you've probably heard this, is biofuels. We're trying to use the microbes as little machines to do work for us, to produce fuel, to answer energy crisis in the future. I mentioned medical aspects. There's also antibiotics. Like I said, there are so many of them, and they're competing against each other, plus they're kind of competing against us. They're developing a lot of chemical warfare mechanisms we could use in medicine. Remediation, again, they're molecular machines. They do a lot of work. Sometimes, in some cases, if you were to clock some of these enzymes, they do 1 million jobs per second. That's on the molecular level. There's no way we can build any machine that competes with that. There's also nanostructures. We find some of these microbes are forming intricate little wires where they actually send electrons along. They're almost like a circuit unto themselves. So these are all the practical reasons why I study microbes. But there is one other reason to kind of study microbes, and this is where I come in. I usually use this slide because it literally, everything I just said is just the tip of the iceberg. If we consider the techniques established by Louis Pasteur in the 1800s, we've only been able to culture 1% of the organisms in the world, in the lab. That's using techniques from the 1800s. That means 99% of the organisms out there, we know they're there, we know nothing about them. We don't even know if they're good, they're bad, we don't know anything. It's not a matter of like knowing something's, oh, well, we decide not to go there. It's a complete void. And in my opinion, that's a bad position to be in. There's probably a lot more good that I mentioned that we could find out. Biofuels, medical, so forth. So, we need to start breaking the old techniques of Louis Pasteur and start developing new ones. And I'm finding a lot of times this, thinking outside, the term thinking outside the box, or trying techniques that seem a little bizarre, actually turn out to be very good instinctive ways of growing organisms. So, microbes. this is an E. coli where it's been chemically treated so that the DNA comes out of it. So you can actually visually see DNA, what it looks like. It's a very large molecule. Now, why DNA is important when talking about microbes is because a kind of revolution has happened in the recent years, 50 years ago, when we were starting to master sequence DNA and find out what is in origin, what the hereditary material that's passed down from parent strain to daughter strain. This also goes on in us, the larger creatures too. So if the DNA can be used somehow to look at microbes in a very specific way, what type of world will that paint for us? Now, DNA itself, this is the actual molecule. So you saw those little strands. This is actually the strand itself. This is a computer graphic, a rendition of it. But it's composed of 4 bases. The bases that you might hear, the rungs of life, or the bases of life. So you have adenine, thymine, guanine, cysteine. In the combination, adenine always pairs with thymine. Guanine always pairs with cysteine. The pattern by which they are found codes for information that makes an organism. It's unique. And so the more patterns you got, the more uniqueness of a sequence. There's a lot of things we have in common. All life on this planet has in common with other life, because to conduct life, you have to have this basic machinery. Now, using DNA, about 50 years ago, a scientist named Carl Woese was studying DNA and wondered, what if, DNA changes, it mutates over time, but what if there's a section in DNA that does not change as much over time? Could we use that to recalibrate the tree of life and understand the hierarchy of where organisms are found? And this is called the 16S ribosomal RNA. It's what we kind of sometimes refer to as a housekeeping gene. All life has it, and if you are living, you do not want to tamper with the basic foundation of life, because then you might turn from living and not living. So, if that's the case, what happens if you arrange all the organisms you ever encountered on Earth based on the 16S? And this is what Carl Woese did. Initially, before DNA, we knew of eukarya, which is where we find us, plants, fungi, and then we have the bacteria, those little animals. Well, within the domain bacteria, there was another group called the archaebacteria, just a name, but Carl Woese found out these organisms are different. Their 16S doesn't line up well at all. So a new domain was established, archaea, which in Latin means the ancient ones. And it's actually kind of aptly named, and I'll go into this why. So the tree of life automatically expanded a little bit just by looking at the 16S. Now, the archaea are arranged in the tree of life in a way that they are actually connected to the branch which leads to us, the humans. There's a reason for this. They employ machinery that's similar to us, and they employ machinery that's similar to bacteria. Why? We're not entirely sure. But again, the arrangement of the DNA on the tree of life puts them here. As the tree of life expands, you see the longer the lines, the more diversity you get. But the base of the tree, the root of the tree, where we believe is the last universal common ancestor, or LUCA. What this organism was, what it is, how it got there, how this even happened, again, we're not too sure, but this is the beginnings of understanding. This is where you want to start to try to map things out. But around this time, again, you're talking 50, 40 years ago, kind of an event happened that kind of turned everything upside down. And it happened here in Yellowstone National Park. For many years, settlers came, the Native Americans were here. People looked into the pools of Yellowstone and saw pretty colors and filaments and so forth. For whatever reason, no one ever considered, wait a minute, if you see those pretty colors and all those things, is that life? Is there something living in the boiling water? We use boiling water to sterilize stuff. How is it possible something could live in there? I mean, look at some of these structures. Later on, I'll talk about stromatolites. This is kind of interesting. Green, that's like a plant, right? All right. So again, This is boiling water. This is toxic water. The pHs are very high or very low. There's hydrogen sulfide. This is not the normal area where you expect life to be. This is one of my favorite pictures. This is absolutely. But again, this is not just minerals. This is a life form in there. So a scientist named Thomas Brock took a sample there and isolated an organism, a microbe, and he named it Thermus aquaticus. It turned out to be a bacteria. It lives in very hot temperature water. It actually gets cold in our ambient temperature. So this was completely new. This changed how we looked at organisms and life forms on this planet. As a scientist, I talk about Celsius. A lot of people talk in Fahrenheit, so I just want to give you this kind of gauge here. So at 32 degrees Fahrenheit temperature where water freezes, this is 0 degrees Celsius. Water boils at 212 degrees Fahrenheit or 100 degrees Celsius. Room temperature, like right now, it's about 25, maybe 22 to 25 degrees Celsius. Body temperature is about 37 degrees Celsius. And the hottest day, this is kind of a recent thing, it's been accredited to Death Valley at 57 degrees Celsius. Pretty hot, right? Some of the organisms found in Yellowstone fall into a category much hotter than this, and we now refer to them as thermophiles. Latin, again, meaning heat lovers. The thermophiles are organisms that live in a temperature between 60 and 80 degrees Celsius. And then yet there's another category altogether called the hyperthermophiles, or the extreme heat lovers, and they live at 80 degrees and beyond. There are some organisms that can survive an autoclave or a pressure cooker, basically. I leave it as a question mark because science is changing all the time, and it hasn't been really a lockdown final temperature where life can live. Like I said, if it was 40 or 50 years ago, no one would have been discussing organisms living in high temperatures anyway, so it's probably a good idea to leave some spots open to new ideas. So if we go back to the tree of life, and we start, again, looking at the 16S and start mapping out where all these thermophiles are found, something really curious kind of pops out. They're all occupying the lower branches. So what does this say to us? This is a suggestion that the earliest life forms, the lower branches of the tree of life, were all thermophiles. Does this make sense at all? So this strange group of extremophiles are actually somehow important to the origin of life. This actually fits in very well with the hot Earth theory, which is the Earth had a very violent beginning where asteroids, molten lava, no oxygen. It was a very violent time, but then it started cooling down. But it didn't just rapidly cool down to this ambient temperature. It started at a very hot water had to appear. So this is an environment where organisms like the extremophiles or the thermophiles can be found. So this is why we start talking about the origins of life. As the world starts cooling, and like I said, these organisms are truly the engineers of this planet because they start converting the chemistries of the planet. These are stromatolites. You can find them in Shark Bay and various, even at Yellowstone. These are complex structures, fossils if you will, organisms that started terraforming our planet, start producing oxygen and other chemicals, basically paving the way for more advanced, complex creatures, such as this cute guy. This is a tardigrade. It's actually a worm, but they also go by the common name of water bear. So this is a more complex organism, and as this other complex organism came to this planet, the microbes are still here, and they're still changing things in the background. So this is one of the stopping points here. And like I said, I just want to emphasize that microbes are everywhere. We have not really officially found any place that we cannot find them. They are and the vast majority of them are known. And the extremophiles, 50 years ago, I still consider that a new discovery. It caused a revolution in genetic sequencing, because the polymerases we use today is the same technique you would see, say, on CSI when they do forensic profiling within genetic material. When you do genealogy, same thing. Even now, there's detergents that you use that have thermal-stable enzymes that will survive the hot water in order to get the dirt off clothes. We use them in foods too. Again, the heat. These enzymes survive the heat. Ours don't. So, and like I said, it's no better way to describe it. They are the engineers of this planet. They do a lot of chemistries that we just cannot do. They just do it very fast, very efficient, and there you go. Like I said, stopping point. Questions right now with what I just, yes?
It's more of like a statement thing. If like we were made out of organisms that could stand extreme heat, we went into fire, we wouldn't burn to death.
Interesting theory, yes. The one thing is that they are kind of dependent upon us still to stay in one piece. So if they were there or not there, our cells are susceptible to fire. They may not be, but they're dependent upon us to stay together. And so it wouldn't work out so hot. Like I said, they would be very sad to have our body gone. Yes?
With the tree of life, was the reason that they separated the archaea from the bacteria, you said because their genetic coding was so different from the bacteria?
Yeah, based on the 16S. These are the housekeeping genes. So when you compare them, and we're not, I mean, you might say, oh, there are massive differences. Well, they're not really massive differences. If you compare to us, it's still DNA. But it's just enough changes that these sequences, if they do not change rapidly over time, then it's like a hard rock to hold on to say, all right, at least we're guaranteed that this is not going to erode or change rapidly in the next thousands of years, so we can then say, okay, yeah, archaea over here, bacteria over here. Maybe in time, the lines will be blurred a little bit. But for right now, they are distinct domains, as we've referred to them in the phylogenetic tree. Again, why that's so, if you look at each domain, you find extremophiles in both bacteria or you find the thermophiles both in bacteria and archaea. There are some things that the archaea do that we thought was maybe unique, and then we sometimes find out that through our gene transfer, those genes are transferred over to bacteria. So they do kind of share a lot of information between each other, but they're just physically kind of different. Sometimes some of them employ really strange lipid layers or armor around us that bacteria don't. So it's sometimes really, if we didn't have the DNA, we would be doing this on visual comparison like the early phylogenetic tree. At least we have DNA to kind of quantifiably separate, do some sort of sorting and separating, because otherwise we got nothing. Any other questions? Yes?
I thought we already knew the beginning of life. First the waves crashed together, and then something happened.
But then what happened when the waves crashed together? Yeah, on a mechanical level, what happened? And at that, if there's waves, there's water. So what came first, water or life?
Oh.
Yeah. So I mean, it's a very interesting question. We shouldn't assume. We shouldn't make a conclusion without proof. So we have to be very good detectives here. Yes?
Where do viruses fit in? Is that like a way left? I know it's a totally different lecture topic, but...
Viruses are not microbes. They are, I was mentioning about enzymes and machines. The best description of a virus is almost that it is a machine. It does work. But sometimes they're bad, sometimes they're kind of doesn't matter to us, but there's a lot of discussion whether or not they are life. Now, they don't reproduce, in a sense, without the aid of a host. So there may be that requirement you might want to make, say, okay, well, you have to be self-sufficient. But there are some microbes that are not self-sufficient. They're symbionts or parasites. So viruses may have come early on in the origin of life. They may have been the predecessor to microbes. We don't know. We do know they do have genetic material, like microbes in general. So they have a placement in the tree of life, but not necessarily like a domain, not like a bacteria or an archaea. This may change. Archaea did not exist until we just made that separation. Maybe there's room to put in viruses. All right, let's move on a little bit. There'll be another stopping point. So the thermophiles. How to study them? You go to the locations where you find them. So I've spent some time in Yellowstone National Park and all the pools. These are, if you've ever been there, it's a wonderful place. You have all these little walkways. You go through the geyser field, Mammoth Springs. It can be dangerous in the sense of just you need to stay on the walkways because some of the waters are like pH 2, it's basically like battery acid, and it's boiling. It's some of the ground is porous, so you have to watch where you step because the ground actually might just cave in. Yellowstone is actually a super volcano. It's a giant caldera, and that's where all the heat is coming from. It's coming from the geothermal reactions below. In this area, you see even the wildlife. If one misstep, because of the bison, and it gets burned inside the spring or into this area. So it is quite dangerous, but again, life lives there. And so we go there to try to catch them. And this area here is called Obsidian Pool. And you can see the water is literally just bubbling, the hydrogen is coming up. It smells like rotten eggs everywhere. Some areas are sulfur bleeding out of the ground, the yellow-orange color. You know, it's quite an interesting place. So, as scientists, we go into the field, we go to pools, we take up samples, put them in bottles, bring them back, and then we start studying them. Now, I'm kind of, I'll say this a couple times, I stand on the shoulders of giants. There are other scientists before me, like Thomas Brock, Carl Stetter. These are kind of the rock stars in the microbial field, where these guys, they went to these areas and they collected microbes and they started kind of laying out like, okay, you need to do this in order to grow them, don't do this, and so forth. One of the main things, these are hot pools, so hot that gases don't really stay in solution. So oxygen is not present. So these microbes are strict anaerobes, meaning that they live without oxygen. Oxygen poisons them. So one of the questions I usually get is like, you know, aren't I afraid to bring back something dangerous from these areas that's dangerous to us? Number one defense is, we breathe oxygen. They don't like that. Second thing is that they are so basic and primitive that for whatever reason, not so much for a reason, they don't have a need to develop any pathogens or any type of nasty interaction with us, because they do very well on their own. They don't need us for the most part. The other thing is that in the domain of life, you got bacteria and you got archaea. Not one archaea has ever been implicated in being a pathogen. Not one. Yeah, you can find lots of bacteria that cause lots of problems for us, but not one archaea. Again, what is that telling us? Not too sure, but it's interesting all the same. This is my bench at work, full of bottles. Like I said, these microbes don't like oxygen, so I have to literally grow all my microbes in these little worlds of different gases and liquids, and this is where they live and grow. And on my bench, this ambient temperature is kind of cold; they freeze. But so I have to take them, put them into ovens, and raise the temperature, and then that's when they start taking off and growing. But this is where you start. I mean, you try and try and try, and there's a lot of failure. Unfortunately, 90% failure rate, maybe 10% success. That's on a good day. But once you start getting something growing, then it's time to start characterizing, studying. What does it do? How does it do it? How does it conduct life? How does it live? This is an organism I characterized from Yellowstone National Park. The names are long, they're kind of hard to pronounce, but they are very important. They give you a description of what they are. So my microbe here is a thermodesulfobacterium geophantus, or the sulfur-breathing bacterium from a heated earth spring. And that picture I showed you, the boiling of water on it, that was Obsidian Pool. That's where this organism comes from. This organism, this is a bacterium. All it needs is all life has to have a certain carbon source. It just needs carbon dioxide. So we breathe out carbon dioxide. Plants use carbon dioxide. But this guy only needs his carbon dioxide, a little hydrogen. Hydrogen's energy to it. There's lots of hydrogen. There's lots of CO2 also. And then it uses sulfate, which is a sulfur compound, to dump its electrons, to get rid of its waste products' electrons. So it needs to get rid of it, so it makes sulfite. That's that rotten egg smell you smell. And so this organism makes all its vitamins. It just needs some heat, no oxygen, CO2, hydrogen, and some sulfate. That's it. You almost can't get any more basic than that. This organism also grows at 80 degrees Celsius. That's the upper limit of bacteria. We have never found one bacterium that grows beyond 80 degrees. Again, an interesting little fact, because the archaea, lots of them actually grow 80 and above. So there is another distinction. So the 16S is telling us that there is a distinction, there is a reason to separate these guys, because there's different niches that these organisms occupy. So, besides Yellowstone, there are many places out in the Pacific Ocean, Atlantic. You have what are called black smokers. Same thing, geothermally heated areas, hot fluids coming up 300, 400 degrees Celsius, dissolving iron, manganese, uranium, all sorts of metals, and it becomes soluble in the water. And then as soon as it hits the cold water, it then starts crashing out as black smoke. But this is the iron, sulfide, manganese, all this stuff is crashing out. And this provides food for the microbes, the extremophiles in these environments. So much so that you'll see big tube worms. We kind of call them sometimes gutless wonders, because literally just that. They have no guts. They're basically, all you see is the red part, which is the filter. They have a sac inside them where they keep the microbes. They use the microbes to feed the tube worm. They know that the high temperatures here, this is a very poisonous, dangerous area, but as long as you make friends with the microbes, the extremophiles in this area, you can live. So, kind of come back to your idea, yes, there are some organisms that do live in symbiotic relationship, tolerating the heat by having microbes around them, but these tube worms, the crabs, the shrimp, they also have other machineries. Their normal cells can take that heat and keep themselves safe, as long as they can also keep the microbes alive. So it is a true symbiotic relationship. So I mentioned Yellowstone, I mentioned underwater, so these are some of the extreme areas. What do you think is probably the most extreme organism that we have captured in the bottle? Like I said, we're talking only the 1% right now. I haven't even gone to the 99% yet. If you want to go and find one really extreme area, it's actually you can visit this place. It's called Volcano. And it's an island off of Italy. I think there's a couple towns there too. And there is an organism I've worked with quite a bit for 5 years of my graduate work. And it's called Pyrococcus furiosus, or the thrashing fireball. It lives in boiling water, 100 degrees. That's the temperature it grows at, and very rapidly. It grows as fast as E. coli does, and that's a record. This organism does not cause any disease. It's actually not even a major organism in the environment, but it's It's one of the ones we were successful in catching and putting in a bottle. Unlike the other one, which is long rods, this guy is a sphere, like little beads. And it has flagella, it looks like a mohawk on it, and that's how it thrashes around and moves around. And this organism is one of my favorites, but at our temperature, 25 degrees here, it freezes to death. So extreme is kind of relative to Pyrococcus furiosus. Our ambient temperature is the extreme. As far as it's concerned, the normal temperature is 100 degrees. So, there are many extremophiles. I haven't gone over all of them, but I've talked a lot about the thermophiles. But there's different organisms that live, oligotrophs, which are kind of the ones I'm working on right now, which live in very low nutrient areas. There are halophiles, organisms that live in high salt. There are all sorts of different environments that we consider extreme, but they live there. And one thing is, it also kind of makes sense, like these extreme environments, you kind of have low competition. Unless you were extremophile, you're not going to encounter anyone else. You're definitely not going to encounter us. So it's actually a good pickings for them to be in those areas. But coming back to the hot earth theory, these extreme environments might have been here before us. So this is actually a very good stopping point before I continue on, so if you have any questions on this section of material. Yes?
How do you get your organisms in the bottles?
The picture you saw me, I'm reaching out with literally a little cup, and I literally scoop up the water, and we put it into literally a glass jar, a reinforced glass jar, put a stopper, cap, and seal it, and then we pump out any air. It's not perfect. Yes, I do get probably a little bit of ambient air in it, but when I'm pouring into the bottle, it's hot, so not a lot of oxygen want to get in there at that moment. And then we bring back the lab and we do what's called enrichments, where I try different conditions that may be amiable to the organisms in there to grow them. There are many, again, this is kind of an old technique. Maybe there's better ones. And some of the ones I've been developing at DRI was to just don't bother trying to collect the water, but push the water through a filter. These are filters that on for camping trips and so forth, you would use to sterilize your water. Well, they sterilize them because the filters collect the microbes, take them out of the water so you can drink it. Well, that's a perfect catching net. So if I catch them all, and then I have to take the filter back to the lab and try to grow them, then maybe that'll work. You know, as long as I keep the oxygen away from them again. So yeah, finding out new ways of capturing them is a big deal. Yes?
Do they die on the way to the lab a lot of the time? Or?
Yeah, they probably do. And again, the numbers, to put things in perspective, like E. coli will grow to the point where you'll actually see it. It gets turbid, smelly, and you can't look through the liquid. We refer to that as turbid or logarithmic stable, stationary phase, or even death phase of microbes. These guys, unfortunately, don't get that dense. And so even though E. coli might get up to billions and trillions in numbers, these guys are only in the maybe 100,000, maybe million range and all that. And yeah, if some of them die, we could take a hit and we may never even grow them. So it becomes paramount to figure out new and better ways of collecting and catching them and not having them die, or you get to your lab as fast as you can and you start trying to grow them, get them into a better environment. So when you see scientists running through the airport with their big coolers, you know what they're doing. Yes?
When you identify a new microbe, do you get to name it?
Yes. If it is a unique organism, you then get the opportunity to name it. So like not Pyrococcus furiosus, but Geophantus, I named that one. So that was one of my, I characterized it. Now, my...
Are you not going to put your own name in one of these?
I'm not famous enough. I'm not famous enough, and also they actually have regulations against that. There's only, they actually had a little bit of a problem. I named another organism after Obsidian Pool and all that, and they didn't like that. And they said, don't, because you might find this organism in another place. That's not Obsidian Pool. So then how does the name help you there? You know, but yeah, no, there are some rules and all that, but you know, there's some room for some creative wording, you know, in Latin, you know. But there are, like I mentioned Carl Stetter, there is a Stetterii out there, but again, he's a very famous German scientist. He's considered the kind of the father of the Thermophiles scientists, so I think he deserves it. So, yeah, no, it's a good question. It is fun, sometimes frustrating. Yes?
So what is the highest temperature they can sustain? Because you're saying that they reproduce the fireball,
Yeah,
reproduce
well,
at 100 Celsius. At what temperature it dies at the highest level?
on that one?
Yes.
Pyrococcus furiosus, beyond 105 degrees, it then doesn't do so well. So its chemistry is very tailored to 100 degrees. It's really right on that edge. And that temperature's beyond that, then it's actually, its lipid structure, DNA starts falling apart. And so, yeah, it's gone. But
does it have the 5 degrees back and towards the colder weather, temperature also?
no, interesting enough, it actually kind of goes into a torpor state, sleep state, below 75 degrees Celsius. And then when you get to our temperature, then they're not doing so hot. They're like almost frozen. It's just kind of good, you know, because as long as you bring them back up to temperature, then you can get them back alive again, but you really shouldn't be keeping them at that temperature for too long, unless you actually put them in a freezer. It's really interesting, and that's actually an area of study that's still ongoing. We would like to think we know exactly why enzymes fold and behave in certain ways at high temperatures, but we still don't. For all our genetic sequencing, for all our understanding of protein folding, we still don't understand why is a protein stable at 100 degrees. You know, we have some clues, but nothing definitive. Yes?
You know the organisms that need the oxygen poison to them, basically, we actually help them because we suck up the oxygen and breathe out carbon dioxide.
Exactly.
But
We
then,
do.
But then the plants, I guess, are enemies to them because they suck up all of the carbon dioxide and turns it into oxygen.
Exactly. We're actually in a symbiotic relationship with plants. They provide oxygen, we provide them with CO2. There's always a stable amount of oxygen in our atmosphere. So unfortunately, no matter how much we breathe, we cannot remove enough oxygen for these type of microbes. And they don't mind, and they don't, because as long as they live in the springs or the hot air zones, the oxygen can't get to them. So, you know, they're not going to go running away from the spring. They're going to stay there. That's their home. Yes?
So if you have an organism that would qualify under several of these, maybe 4 or 5, like they live under rocks and in high temperature and in high pressure. I imagine at the bottom of the ocean there are things that are high pressure, low temperature, low nutrient, no sunlight kind of things. First of all, how do you name those? Is there like one thing, is that a thermophile or is it a piezophile? And you qualify it that way. And what is the organism that you found that lives with the most difficult conditions total?
The naming scheme can be a little bit arbitrary, but usually it's focused in on its greatest attribute. Okay, so yeah, an organism can share lots of different extreme characteristics, but usually it's kind of well known for one characteristic above and beyond others. And we kind of use that as the guiding star as to how to name it. Because again, we want to be informative of what this organism is, so I might as well use its best trait to describe it. What organism has the most characteristics? I'm not actually sure. I'm thinking of right now, there are probably 5 or 6 of those characteristics are about the most I've ever seen in any given organism. It's not to say that you can find one with more. There is an organism I will introduce you to that is kind of ranks up there in the 5 range. It's pretty bizarre. So I'll get there. Any other questions? Yes?
So you said that the thrashing fireball grows rapidly at 100 Celsius.
Mm-hmm.
So would it still grow like, let's say, 80 Celsius, but less quickly?
Yes, exactly.
Oh.
Yeah. We call that a temperature range. We'll say a temperature optimal, but then some of these organisms will grow at lower or higher temperatures. We're not terribly happy about it, but they'll do it. Some of the microbes in our mouth, interestingly, we have a low pH in our mouth and all that. A good friend of mine studies these microbes in the mouth, and he isolated a few, and strangely enough, they grew better at a higher pH than a lower pH. Now, the saliva in our mouth is a low pH. So it's on purpose. We're trying to control microbes. We don't want them eating our teeth and all that. But if you raise the pH, they do even better. So yeah, just because you find an organism in an environment doesn't mean that's its optimal. It just means it can be tolerating to live there. You know, waiting for an opportunity to do better. So, all right, moving on. So I was talking about the tree of life and the discovery of thermophiles. That's about 50 or 40 years ago. At the same time, another big event happened. The USA landed on Mars successfully. Mars has been very challenging. About 50% of the satellites and landers have failed. Russia and the US have tried really hard. Viking 1 and 2 were the first continuously successful landers again on Mars. Started returning wonderful pictures of the landscape and we started doing our first studies, chemistries of this planet. This is another planet besides Earth. I'm going to use some updated data, but the Vikings found the same thing, and that is Mars is kind of cold. So temperature ranges from -90 to a balmy -3. It has lots of carbon dioxide, lots. Its atmosphere is very thin, but around 95% of it, or 96% is carbon dioxide. There's some argon, some nitrogen, very little oxygen. There's still some oxygen. Carbon monoxide. This data was collected by Curiosity. So when we landed there, we didn't know about extremophiles. I mean, it was these are two concurrent thoughts happening at the same time. So it's really kind of hard to say. Maybe NASA at the time, their scientists probably were keeping up on the literature. Like, okay, wait a minute, it's like we just landed a lander on there and we're going to be doing these. And wait a minute, there's these extremophiles. We should have been looking for them. This happens lots of times. We have to just gather information as rapidly as we can and react as efficiently and rapidly as we can. So Viking had an experiment, the first very ambitious experiment, which is a robotic arm to scoop up some dirt, bring it into the lander, and then sieve it into 3 different containers, and where they incubated, added some water, put some nutrients in there, incubated, and detect life. They used isotopes, stable radioactive isotopes in the nutrients so that they knew what went in, and if a microbe is going to consume it, it will then kind of breathe out CO2, like E. coli. This experiment was done on both rovers, and both experiments came back with the result that a stable isotope of carbon was produced. There was life in the soil of Mars. It was quite controversial. This is one study done on 2 landers, but it was very promising. And we're looking for standard life that lives in our ambient temperature. We like to build upon results that are reproducible and point in the same direction. A little bit later, Phoenix, another lander, came back and found in the soil there was a molecule called perchlorate. This molecule is stable at cold temperatures, but in the Viking landers, we heated the soil up, thinking that you needed heat to get the life forms going. When you heat this molecule up, it starts reacting with everything else and producing CO2. So the result is inconclusive, because the result could have been the perchlorate, not necessarily life on Mars. So a little bit of a disappointment, but we are learning, always learning, gathering more information, finding out how the soils and how Mars behaves. So Mars was starting to look like, for a while, they're a little bit lifeless. That no, life is not on it, at least not on the surface and so forth. But about 10 years ago, another change in our understanding in microbes happened. 10 years ago, a discovery was made in South Africa. Now, my mentor, Dr. Dwayne Moser, was a postdoc at this time, and he went to South Africa and went into an African gold mine. 3 kilometers underground, they hit water. Water came up into the gold mine, and then they called in the scientists, because this was kind of unusual. You're 3 kilometers, almost 3 miles down here. They tested the water was coming further down, another 2 kilometers, so a total of 5 kilometers below surface. The water was coming up, and he looked into the water coming in, and he saw, and there's some little pictures here of the actual mine. This is the shaft going down into the mine. It's so deep you can fit, I believe it's 8 skyscrapers in it. This is the actual tunnel. These are pipes that pump air in there so you can breathe, because it gets very hot. There's a lot of methane, carbon monoxide. It's not very friendly to humans, but a potentially good place to find a microbe, especially an extremophile. So in the water was something strange. That looks like a cell. This is an organism that we came to name. He named it Desulfurutus aurochs viator, ore, a bold traveling sulfur-breathing rod. Audax Viter comes from Journey to the Center of the Earth and Jules Verne, so I thought it was aptly named. So this is an organism that's found 5 kilometers underground, and it's the deepest organism ever discovered on Earth. So, like I said, there's no place on this planet, so far, we have not found life. So maybe at 6 kilometers, maybe that changes. We don't know that yet. This organism, like the Geophontis mentioned before, is a very basic life form, but this one does something a little strange. Down there in the deep, there's no sunlight. There's no input from the surface. There's just rock, water, and radiation. Natural radiation. All planets have uranium, so it's natural background radiation. It is hypothesized that the radiation could react with water, producing hydroxide and hydrogen. Hydrogen, like I mentioned about Geophontis, is an energy source. The hydroxide can react with iron sulfide and generate sulfate. It could also react with the carbonate, making CO2. So this organism could live independent from life on the surface, on a very minimum lifestyle, such as this. So the dinosaurs come and go. Ice ages come and go. We arrive. This organism doesn't care. It lives separate from the world up here. And it was the only organism found down there, which is highly unusual. And we find diversity everywhere, but down there, only this thing lives. It dominates the subsurface, at least in South Africa. So this organism, rock, water, and radiation, is very basic in these conditions you find in other places other than Earth. So, where do you find this other besides South Africa? You find it here. That's what we're doing here. We're starting to seeing organisms. So Nevada Test Site, or the Nevada National Security Site, is one location. We also are in South Dakota in what's called Surf. It's another gold mine. You can see right there, still gold in the walls. And microbes, extremophiles, are living there. There's even you find ash meadows in Amargosa Valley. This is BL1. When you go driving, you might see little houses with solar panels or these little red boxes. Those are wells. To us, these are wells that may supply water to you, but to us, these are windows to the subsurface. We just pump up water from below. We get to take a look at what's going on down there. There's a great boiling spring in northern Nevada. All these are locations you go. You don't have to go to the exotic Yellowstone or Kamchatka, Russia. These are homes to the extremophiles. Now, unless you take a water sample and look under the microscope, you won't necessarily see them there, but you definitely can smell them. There's a lot of rotten egg you can smell in there. Now, you're mentioning about an extreme condition where multiple things kind of come together. This is one of my organisms I'm working on right now. This is so early in the stage of this organism, I don't even have a name for it. Right now, we just call it DRI-14. This organism is very small. Most organisms are about 1-2 microns long. A micron is a meter chopped up 1,000,000 times, and that's a micron. Or a human hair is about 100 microns wide. This guy's about half micron long, so it's very small. It also floats, which is kind of annoying because I didn't realize it was there. It floated literally away. But this organism is very special. Well, all your organisms, every time you discover one, they're always special, but this one kind of blew my mind because I tried a condition that was odd. Because down there in the deep, all you have is water, rock, and there's usually hydrocarbons. One of the most plentiful in the universe is methane. This organism can grow anaerobically without oxygen, rock, water, methane. It can also use hydrogen and CO2. That's actually when it's really living good here. That's when it starts growing. A lot of these organisms, like desulfurous, they're slow growing. That's why when I mentioned dinosaurs come and go, ice ages come and go, that's their lifespan. And so you're not talking about days, you're talking about eons, you're talking hundreds of years, thousands of years. We really don't know how they pull this off. But they don't live fast. If you live in the deep, you live slow. And that's how you preserve yourself. This organism, rock, water, and methane, is similar to desulfurous, rock, water, and radiation. And they are actually related to each other by 91%. This is by the 16th acid yet. You think 91% is not really related? Well, in this category, there's only these two. We don't, again, back to the 99%, we don't know who else is in this area because we never caught them. So it's a place to start. So, rock, water, and methane. Where else do you find those conditions? I'll give you a hint. Mars. Yes, Titan does too. But Mars has methane. It's, and besides methane, the soil of Mars is similar to Earth. There are some differences, but it's similar. There used to be water there. We detected methane in atmosphere. This is actually still kind of controversial because the satellite detected, but the rovers haven't. So it's like, who's right? Is there methane there or not? But if we look at other moons and planets in our solar system, methane is there. So it's not really unusual to think that the methane is not there. So we've gone back. We have brought Curiosity there. It's a very large rover, has many different sensors and equipments, sophisticated equipments to detect conditions. We're not maybe being as ambitious to go after life like Viking did, but it's not a matter of they're not interested. They are very interested. It's just that we have to learn as much as we can. Again, 50 years ago, we didn't even acknowledge extremophiles lived and how they lived. So we need to find out as much as we can, in any way we can. So every time a successful rover gets onto Mars, that's a win. We can then pull in as much data as we can and find out and to get at the answer of life. Now, the surface may be very uninhabitable, but again, on Earth, 5 kilometers underground, we have life. What if we went underground in Mars? We could find a desulfur Brutus or a DRI-14 on Mars. It's very possible. Problem is, it's very hard to get a drilling rig on Mars and not have humans. We need, robots are great, but sometimes we really do need a human touch. So that's definitely a tug of war that's going on right now. What do? We put humans there, or we continue with the robots. Either way, the robots are very sophisticated. They take selfies of themselves. But you look at that, and you really can't necessarily say that's entirely different from, say, Las Vegas or any other desert. We may consider it an alien world, but really, how alien is it? And we always expect when we find life, it's going to be alien, it's going to be different. But how different? What if, when we do find life, and we sequence it, and assuming it has DNA, is it going to be really different? It might actually, we might be able to rank it up here. It might be actually related to us. It may be down here with the bacteria or archaea. Or, we might be looking at something like this, the origin. But we don't know. We haven't found life yet. We need conclusive data to get there, to build up our repertoire, our understanding of life on this planet and elsewhere. So, why study microbes? Why study extremophiles? This is Earth. This is where we live. This is our only home. It's the only place where life we know exists. This mote of dust held in a sunbeam. As a scientist, we stand on the shoulders of many other scientists. And I come from a really great lab, and Dwayne Moser is my mentor and PI. We have graduate students, undergraduates in our lab. We have high school students too. We do real science. We produce real data. It's an emphasis that this, we do teach, but this is the real deal. All right. We have many collaborators, many former members, and as scientists, we depend upon funding. Science is not free, and times have ebbed and flowed over time, but through NASA, through DOE, DRI, science progresses and we continue on. And my talk's done. And any questions you have now? Yes.
So, I think I've always had this topic on my mind because I wondered the question about life on other planets, and I wondered if, just like how the microbes evolved and adapted to the conditions on Earth in its various stages, why couldn't it have happened on, say, Neptune or Mars?
Exactly.
Or any planet?
That's the exact question we have, because like I said, the Extremophiles really did change things up. We did not appreciate that life could live in such extreme environments. And with that knowledge, we now start looking at other planets and seeing, yes, why not? Why not? And it really kind of gets at, like, how prevalent is life? Again, it's like, we don't know. We have only one data point, Earth. That's it. Yes?
If we do find other species, I hope it doesn't end up like Halo.
Well, I mean, it would, think about this, if there is a life form you encounter and it's big enough and large enough that you actually identify it as, like, you know, whether it's the size of a rabbit or the size of a bear or whatever and all that, at least you know it's a life form. Unfortunately, typically, it's probably not going to happen that way. It's going to happen very small. We may already be encountering them right now, and we never know it. You know, like I said, 99%. That's not a good position to be in. Yes, there's a lot of benefit to knowing more, again, medical and biofuels and all that, but it just means that we know so little. Yes?
I came in a little late, so I might have missed this part, but how did you stumble into this area of study? Did you start out looking at dirt and then look up there? Or did you start out looking up there and then it was like, how do I study that more? And then they were like, well, here's this cup of dirt.
Well, I would say I started off as a child watching Cosmos with Carl Sagan. I also watched Jacques Cousteau. And when I first saw a black smoker underwater, when it was discovered, my imagination was ignited right there. And I saw, I might not be old enough to go to another planet, but that was another alien world right there by the black smokers. And then you have Yellowstone and other areas, and you look at these places, and they look, you know it's Earth, but it's so different, so strange. And then I started investigating, reading a library like this, you know, reading books, trying to find out what is known. Now, you know, I was born much later than Carl Woos when he made his discovery, so Extremophiles were already kind of discovered and moving on. And so I started learning about it. And then I, you know, went to school, went to college, got good grades, pushed on, and suddenly realized, well, I need to learn how to work with these guys. And so what professors out there, who is working on it? And again, go to a computer, go to the library, look at papers, find out the articles, who's publishing, who's doing what. And so I applied to a very few amount of colleges. And all those colleges had professors who were working with the Extremophiles. There weren't many, I'll tell you that. There was like maybe half a dozen. And I got accepted at the University of Georgia, where Dr. Adams, and he worked with Pyrococcus furiosus, the thrashing fireball. And that's where I started my career working with the Extremophiles. And Pyrococcus is pretty forgiving and easy to grow in the Extremophiles. It's still pretty awesome of an organism to work with, because everything's boiling and you can for everything. But it got me interested in other organisms. And so as I was developing my research, I was comparing, we were using a technique called microarray, comparing Pyrococcus to other organisms. And for my PhD, I discovered that there was another organism called Pyrococcus luzii, after Carl Woos. And strangely enough, you look at the 16S, are identical. So like this one blew a hole in Carl Woos. They're like, wait a minute, they're the same. So aren't they the same? And I tested them out and grew them, and no, they behaved differently. They grew differently. So then I genetically compared them and found that there were distinct sections in Carl Woos that were missing. Furiosus had them. So what I accidentally found was pretty good evidence was called lateral gene transfer. The transfer of genes, not by vertical means, not by descendancy, but by stealing or sharing them with another organism at 100 degrees. So this, again, feeds into the idea that the origin of life might have been hot, it might have been warm, And that it might not have been individual organisms, but really a collective sharing of genes, kind of like trading baseball cards, you know, and good combinations made robust life forms. And they took on. So that was my introduction. I was hooked. and I will never go back. This is, my Extremophiles are my little pets, my family, and it's always fun seeing what they can do, you know, and having them teach me what to think. Sorry, long-winded answer.
So when you say things grow, are you saying they get bigger? When you grow things, you're making them bigger, or are they replicating?
They're replicating. Yeah. Bigger is kind of, again, abstract. They come in different sizes. Micron scales usually the area they're in, but you do find some exceptions. There's an archaea that I think is called gigantus something. It's actually big enough you can see with the naked eye. In theory, it's like a particle. You know, that's an exception. Most of the time, you never see them. But yeah, when we refer to as growing or cultivation, we want more of them. We need them to grow to numbers of, from, in theory, 1 cell should make 2 cells, 2 cells should make 4 cells, and so forth. But again, E. coli, trillions upon trillions of cells. These guys are lucky they get into the millions. And that's maybe another reason why they remain invisible, because there are just not many of them. Doesn't mean that they're less important. They just don't get into the numbers necessary to be like an E. coli. Yes.
So I hear all the time like the big time scientists always mainly are saying that there's no way there could be life, and there's the other sense, but it's like smaller amount of people saying there's definitely not be life, because I always think that it doesn't have to stick to our exact like chemistry of how life works, and on other planets that we haven't even discovered yet, there could already be like whole like civilization of sentient beings like us that didn't evolve like us and didn't follow the rules like how scientists say.
Well, there is a certain set of rules within chemistry that are kind of hard to break, but within that, you see a lot of diversity. Dinosaurs used to be here. They're pretty different. They're not here anymore, but why not say somewhere else? As far as, you know, when I was going through grad school, I remember it was me and one other student, and we were so excited when a paper came out and they were talking about an idea that DNA and protein could survive inside an asteroid after impacting on Earth. And we're like, wow, this is so cool, this is interesting. And other students and professors kind of laughed at us. It happens. You know, but, you know, science is based upon reproducible results. Results that you can bet on of happening because you now know the rules by which it runs by. If you can find those rules, testable rules, then you have a science. You have something that is workable. Now, as far as life doing other things, of course, we're always learning. Again, if I told you 50 years ago there's an organism that could live 5 kilometers underground, would you believe me? No. You would think I was a crackpot. But here we are. We're changing. We're learning. We're relearning the rule book. And that's fine. That's what science is all about. You really have to be open to strange things. I tell my students all the time, they'll say, this can't be right. You know, they're growing this or they're doing this. And I'm like, who says what is right? You know, us. What do we know? You know, let them tell you what to think. Then test it. Test it again. If it's still the same result, then publish it. You found something that at that moment in time, no one else knows. Yes?
I have a question about cold temperatures.
Mm-hmm.
So is it true that in the cold temperatures, no organism, extreme cold, the organism either die or go to sleep, and there's no organism that reproduces at extreme cold temperatures?
Well, again, at DRI, there's one of our scientists is looking at microbial life in the frozen lakes in Antarctica, and there's life there. So even though it's freezing, now this is salt water that freezes. Most water will form a clathrate structure, expand, and turn into a solid. But if there's salts and other stuff dissolved in there, you get these micro channels of liquid water. and it's liquid water, and so life can actually live in there. So this is a combination state where you have a psychrophile, cold lover, and a halophile, an organism that loves salt, wants lots of it. Makes perfect sense, because the water freezes, but the salts stay liquid in between the cracks. And this organism lives there, reproduces, does fine. So you probably could get to a temperature, maybe, if you go to negative 80, negative 100, really go, you probably will eventually freeze everything, and no life will continue. But interesting about cold, you don't really kill, you just hold an organism in torpor state, a sleep state. With that said, to live in freezing temperatures, you have to have a different chemistry. So I don't know if anyone has ever heard, there are in the Antarctic and in the Arctic is an organism, it's called an ice worm. I don't know if you've heard these guys, they're really curious. They're purple, and trappers and so forth reported saying they saw worms coming out of the snow. And again, people said, you're crazy, you drank too much or something like that. No, that's not possible. Scientists went out there, cut out a big block, sat there, waited for the sun to go down, and eventually worms started coming out, purple worms came out, and they were living in the frozen ice, and they came up to the surface to eat algae and all that, and so they kind of lived in this little twilight time between the sun up and the sun down, and they go back into the snow again, and they stay down there. When you pick up one of these purple worms and hold them in your hand, now you're a blazing 37 degrees Celsius. The worm melts in your hand because the chemistry, its lipid structure, is geared for frozen temperatures. We're just too hot for it. So there's where life has found a way, in a chemical way. It's obeying all the rules that we obey. It just tweaked it a bit. So I'm glad to answer the question. There are psychrophiles and lots of different things. Yes?
So have you encountered any extremophiles that are, I'm trying to think if there could be life inside like gas giants. So if they like high pressure environment, high or low heat, I guess, and also a ton of whatever's in the atmosphere, methane or hydrogen, and they meet up with all of those things. Is there anything that qualifies with that description that you've encountered?
Yeah, we actually have them here on Earth. Again, DRA has monitoring stations in Colorado, and we literally collect clouds to sample the clouds themselves. And in those samples, microbes are found. So microbes drift in the upper turbulence and travel across the world. So they do it on Earth. Why not on Jupiter? Why not on Saturn? Why, again, there's not one place on this planet that we have found yet that doesn't have life, including in the clouds. Yes?
Another DRS science note was Dr. Lynn Margulis, and I think she is also a microbiologist.
Mm-hmm.
And she suggested in her talk that if something catastrophic should happen and all human life forms went away on the planet, she says she imagines the microbes marching over the hill saying, we got along before, we bet you will get along after along, but they will. That's what will populate and re-energize the Earth.
Very likely, but I mean, in a sense, we have a connection with this planet. All life does. Like I said, life is only found here. The microbes have their place, and like I said, they are the engineers of this planet. So if something were to happen to us, life will continue on, but it wouldn't stop. Something else would come about. Maybe another variant of us. Like I said, lateral gene transfer occurs. So innovation is alive and well within the genetic code. Like I said, it's something that robust, that powerful, if you will. It's really hard to believe it's only here. It has to be in other places in our solar system. It really does. Because life is infectious. You're really hard pressed to find a place where it's not there. Yes?
Do microbes talk?
They do, in a sense.
Oh.
It's called quorum sensing. They actually communicate by chemicals. And there's a lot of interesting chemistry there, because how do they know when an invader is there? They can't know with eyes, but they know chemically they're there. How do they know when, say, we're ill or if this is a good place to live? Again, they chemically, they kind of taste the environment, and they, on a mechanical level, they make a decision and they grow and so forth. So yeah, they do talk. It's just not the way we do. Yes?
So you talked a little bit about the difficulty in trying to sample when you're out in the field and you're sampling something, you're trying to bring it back and grow it in the lab. So is there a field of study that's looking at what these microbes do when they die, or when you pull them out, let's say, out of Yellowstone, and you can't get it to grow again in your lab, and you're looking at it? What does it just decay? Does it disappear? Can you see it a year from?
You can sometimes, like when an organism dies, just like it gets decomposed, the materials kind of get spread out. But one thing with microbes, again, the genetic era is now. Genetics is a trace. It's a leftover, if you will, of that organism. So even if the organism dies, we can actually harvest the DNA out of the sample and get an idea of what it was or what it is, and maybe find out why it died. Desulfuridus, for example, the one that came from South Africa, we've never been able to grow it in the lab. We know it exists because we pulled the DNA out of the sample, and that's how it. Now, DR14, I have it in the bottle, and I'm now in the process of pulling the DNA to try the sequence. So you're not guaranteed one way or another. I mean, sometimes you find the organism, you grow it, or sometimes you find the DNA, and then you try to grow it, or whatever. It's based at this point, we will take anything, any type of little victory, we'll take it. But yeah, the leftovers, the lipids, the DNA, all these are tracers of organisms. They're all unique. That's why the archaeous bacteria, the lipids are entirely different. The hyperthermophiles, like Pyrrhococcus has what's called a tetraether membrane, meaning that it has one molecule stretching across its membrane to hold it together. That's how it survives at 100 degrees. It's like discovering a nuclear-powered aircraft carrier in the Stone Age. It came up with a means to survive the heat. Bacteria don't have it. They, at least their lipid layer, cannot survive that heat. So every little trace, even when they're dead, is a signature for us now to find out how they conduct life. That's another thing on Mars. If we can find even that, that would be wonderful. But unfortunately, it has to be distinctive. It has to be kind of without error of doubt. Then we can start approaching the idea that there is life on it. Or until we get someone actually stepping foot on Mars and bring a microscope with them and dig up the soil, put it on there, and we can say, okay, yeah, that looks like a cell. Until we can do that, we're still guessing. Yes?
So going back to the tree of life, and you also mentioned that there was one of the theories that there was an exchange, genetic exchange. I don't know if it's one of the theories or the theory. Mm-hmm. Forgive me if I'm expressing myself. I'm not a scientist.
No worries.
But that it was exchange, a genetic exchange, mm-hmm. to create whatever life we have right now. So what is there like a major idea accepted? Was there one organism that started all, and then lately, whatever you study different kind of, it's just, they changed as adapted to different environment? Or what is there a way of finding out?
That's like $1 million question right there. There's different variants to that. So I was mentioning about my work with Pyrrhococcus and lateral gene transfer within the thermal vent. That's actually kind of supportive of one of the hypotheses, is that to think that there was one organism that spawned all life on Earth may be a little bit too much responsibility for one little organism. So maybe it was more of a collective, that saying it was a species or one organism is too much, but if it was a interactive collective, a library of genetic information that existed, and then eventually life forms crystallized out of it, that's how life started. There's also the idea, okay, if it was one life form, does it have to start here? Maybe not. Maybe it started on Mars. Maybe it started somewhere else and then came here by a transport such like an asteroid. Maybe life did start here. But again, we know the rules of chemistry. We just don't know how it is connected and what the final answer is. Maybe there's not necessarily a final answer, but we got a lot of good hints. And again, like I said, it's just a matter of good detective work. We have to piece things together in a very logical, methodical, retestable method. There's a paper and a very famous event called the Miller reaction, where this graduate student took a glass ball, filled it with gases that they thought was the primordial soup of Earth, put 2 electrodes in it, and then shot an electron bolt through it, like lightning, and all that. He left it for a month, came back, some black soot, yucky stuff came off, and they tested it. They found amino acids in there. So, what was originally kind of thought was a stupid idea turned out to be a clue that maybe you can make amino acids or DNA without anything special other than gas and electricity. So that's, again, a clue. What if the next experiment conducted by one of these young gentlemen here might add one extra thing to this, and we put the puzzle together. What was really interesting, when this graduate student presented his work at his university, he was, according to what I read, he was very nervous and he was hitting a lot of trouble, a lot of resistance from other elder scientists and all that. At the back of the room, when he gave his lecture, there was this other young scientist who was very attentive, very interested in his work. Some strange name, I don't know, Carl Sagan. So it was a great story. Yes?
Can microbes be used for terraforming other planets?
Yes. I mean, we have pretty good proof they've done that here. So that's one of the main things. Every time we land a lander on Mars, we're really, NASA's very worried about us bringing Earth microbes to Mars. They have to go through space. It's unshielded. They land on the surface. It's being bombarded by the solar wind. It kind of goes through its own sterilization process on it. So, at least for now, it is concluded that any microbes that could have been brought there will probably blast out of existence once they get on top of Mars there. So, it's not to say it's not possible, but yeah, once we're kind of satisfied, we found out what we want, and if we're really serious about it, we could go to Mars. We could start putting a colony there. We then would start, rapidly introducing things that we're trying to curb here. We would do a lot of it over there. Bolster that atmosphere, make it thicker, put some oxygen, put CO2, put methane in there. Do the greenhouse effect on purpose, and yeah. The microbes would then hopefully take over and start working away at it. We're talking slow only because you're talking about a big planet. Mars is so small on Earth, it's still big. So it's Earth, human lifespans are small compared to the microbes. Not so much because of their individual life, but because of what they do. They do things on a grand scale. There's a new group of organisms in Archaea called Archaea. They actually cycle almost seemingly now all the nitrogen in all the oceans. We didn't even suspect this. So they are a major nitrogen cycling organism. Again, more of them than there are us, and they're in control of that cycle.
I think I know a way so it can clone us. All we need is the gas container and the electric stuff, put it in a machine that can we can take a sample of ourselves, put it in a tube thing that leads to it, turn it on, and maybe it will clone.
Are you familiar with a scientist named Craig Venter? And he actually developed a new method of how to sequence DNA. And he did it better and faster than the original plan by the U.S. government to do sequencing sequencing of the human genome. We pretty much have to thank for all of the advanced technologies because of his innovation. And so, yes, we actually could clone humans if we wanted to, but it's a matter of an issue that we're still on the edge of knowledge. I was mentioning about microbes in our bodies, we don't even know what they're doing there. There's a study that the microbes actually might control our behavior, which seems really bizarre. But they tried it with mice, and they turned mice that were normally very passive into very aggressive, and very passive microbes very aggressive to very passive mice by changing the microbes in them. That's really strange stuff. We're just learning. So again, baby steps, one thing at a time. We'll get there.
Like, the problem with cloning is because when you say it's inhumane, like toying with, I can't remember, but, toying with, like, nature and stuff, like, when God's work and stuff, but I hear lots of stuff like that.
Good control is always necessary no matter what you do, whatever you do, okay? We are learning something that could cure diseases like cancer, Alzheimer's. There's a lot of good that can happen about this. Yes, maybe some bad can happen about this, but there's no, per se, rules other than trying to figure out what the rules are. We just don't know. We really don't know. We might be able to change things for the better if we just knew better. But, like I said, at 99%, we know so little. So right now we're still learning our ABCs. When we get to sentences and paragraphs, then maybe we should do a little bit of evaluation. But even then, it's a pursuit of knowledge, and there's usually no end to it. But at least points that we could strive for. That's it. Oh, yes, sir.
Just one last thing. It seems I've always called the theory of taking a cup of water and looking at the cup of water the cup of water theory. It just seems like there has to be a better way to research this kind of stuff without glass bottles in a lab. What things like the filter are coming down the pike for better ways to study in these extreme conditions?
There are some methods where they use electrodes. So we are electrical on a chemical sense. So what if you provided an electrical environment where you gave them energy on purpose by electricity? So that's another way you can try to capture them, if you will. There's things now that are working on NASA where they literally drill a hole in the ground and they put in a whole cartridge, which then samples water and nutrients as it passes through the earth. This takes years, decades even. You even introduce rock for them to colonize, and you pull the plug out and you look at it. There's lots of different ways. There's also, you can do, if you know a little bit about the organism or the thing you're looking at, you can then sequence it. You can then make more of it in the lab and so forth. There's a problem to that. Just like D-sulfurous, we sequence the genome. You think we know the genome, we know everything about it, right? We can grow it, right? No. I can't do it. I have tried everything right now. So it just tells me that there's something else there, even though I have the book and I can read it, I just don't have the spirit of the story to get the organism to go. The question about do organisms talk? They do. What if I'm not speaking its language? Maybe I need to have something in there to make it, to signal to it, times are good, you need to start growing. But it's a chemical thing. You're not going to find that in the genome. That might be in the environment. So then you're kind of almost still stuck going, get that cup of water, because then you start analyzing what's in the water. Is there anything in there? Something besides the life form. So, like I said, we know so little. Sometimes, it's a great place to be because no matter where you turn, it's something new, and it's great. But it's also frustrating because it's so new, you can't reference anyone. You can't talk to. You can't say, hey, Bob, whatever. When you did this, what happened? Because Bob didn't do it. You're there, out in the front, going, oh, what did I do? DRA-14 is one of those guys. And I reported to my mentor, Dwayne, and said, I think I got something growing on methane. And he goes, that doesn't happen. Yeah, I know. There's no chapter in the biology book, microbiology book, that describes that. Doesn't mean it doesn't happen. Now I just have to be diligent and map everything out as best as I can. As smart, hopefully I can be smart enough to outsmart the microbe. Yes?
How do you know how old a microbe is?
Very good. Very good. Okay. When you characterize rock or water, you look for what are called isotopes. atoms that are radioactive but are decaying at such a slow rate, you can then determine the age of it. Now, that's in rock and that's in water or anything. Well, that has nothing to do with microbes. Where does the microbe get its material to be it? We eat a candy bar to get the carbon to make more of us, or more cells. Where did that candy bar come from? It came from sugar, chocolate. Well, where did that come from? It came from a plant. Where did the plant come from? The plant got its nutrients from the ground. You're going back to the earth, you're going back to the gases on. So if you knew the age of the atoms in the soil and the earth and the gases, and then when they get uptaked into the microbe, into how many percentage-wise, how much of it is in there, you can then determine the age of a microbe. Most of the time, the microbes are very young and relative to their environment because they're turning over very rapidly. But in the case of Desulfurutus and other organisms, they're growing so slow, we're not entirely sure. If we go through all our calculations, they may divide like once every 100 to 1,000 years. And that's the best it gets right now until we find out something new. But that's slow. E. coli doubles every 30 minutes. Pyrococcus doubles every 30 minutes. We refer to that as doubling time. The doubling time of Desulfurutus is like maybe 100 years or 1,000 years. The doubling time of DRI-14, right now I'm clocking in at 36 days. So one cell becomes 2 cells in 36 days. You have to be plenty patient.
What about in places on Earth where the environment changes quickly, right? You have a new environment introduced, like think of what happened in Fukushima in Japan. Are people watching that to see if, in areas where the water is superheated, if these lives, if these organisms show up, or? If
they're not, they should be. And this is where, this is a very fundamental question. Change happens on this planet all the time. A volcano erupts. All right, so some instances like Fukushima where the humans cause something. Just the general idea of what happens, the rise and fall of empire of microbes. What if no one supports this type of science, we'll never know. And I'm not saying it's necessarily entirely the fault of funding or scientists. It's just that fundamental research develops into applied research. Applied research develops into discoveries, which makes our culture. Something like that, we need to know. Because we don't know. Yes, change, on a basic level, change does happen. When you introduce a new nutrient, a new heat, or take heat away, microbes, like I said, empire rise and fall. The E. coli will leave, the Thermophiles will arrive. And they'll take over and colonize the area. How they do it, why they do it, in what fashion, we may not know. But again, it's worthy of study, because we might learn something that we just didn't see before. But, if I just assume that it happens, then that's good enough, right?
One thing I do know is that every step we take, like, for instance, if I left my drink over here, walked up here to get it back, we kill stuff. We're all mass murderers. So we kill a whole bunch of the micro things.
Yeah. Again, life on this planet, we're integral to each other. Einstein said if the honeybees were to disappear, we would be in terrible danger. And this is very true. In the sense of that, yes, some organisms will die. It's a recycling of material. It has to happen, if you will, but just because it happens doesn't mean it's necessarily bad. But if an organism that's integral, a key member of some chemistry or something that's on its way is removed rapidly, and no one can come in and replace and take over or take up the duties, that can be very dangerous. So again, life is very good at adapting, changing, and getting into places that we thought wouldn't be possible. A lot of times we kind of consider, like, is it detrimental to humans? Well, maybe it is, but in general, life will continue on because it's very innovative. it's really hard to kind of stop it. So I guess my general answer for you is that, yes, your microbes right now are killing off old invaders that want to come in and make you sick. But they're doing you a favor because they want to protect you. You're their home. So in that sense, it's a good thing. But if your defenders were then to suffer a blow and then invaders get in and you get sick, then that's bad. So ebb and flow of empire. The microbes are here, they're not all bad. They're good. Some of them.